Hey, sorry guys, I'm a little bit late today. Not entirely sure what happened there, but I seem to have slightly lost the time and for some reason didn't connect. But not to worry, I am here. No need to panic. Nice to see that you guys are flirting again on the comments. Enjoyed seeing that. Hopefully you guys are all looking forward to doing some protein stuff and recapping hopefully what proteins are and everything else that you need to know about it. Sorry that was a bit late. That was not really my fault, but not entirely not my fault. So we'll leave that there for a sec. I'll load up what I've got in front of me so that you can see stuff and then we shall get started. Okay, hopefully you can see that. Let's just check. Lovely. Okay, cool. Hi guys. Nice to see that you're all here. Anyone saying anything interesting? No, someone's just failed a test on this. That's ideal. What a good time to be doing some revision of proteins today. Right, so what we're going to do today is we are going to be looking at proteins. So we're going to try and cover a whole load of things and I'll tell you exactly what in a sec. Okay, in case you haven't done any of these webinars before, in case you're new to this because someone's told you about it and you're hopefully I don't know, on the receiving end of someone banging on about this. I'm the head of biology here at SnapRevise. So my name's Oli. I have a bachelor's degree in biology. So I went to Southampton University a few years ago now. And I then went to UCL for a year after that. And since then, I've been teaching A-level biology in a secondary school, which I've been doing for about 4 years. So hopefully I'm one of the best placed people right this second to be teaching you some stuff about proteins. Hopefully I'll be able to help you get to the next stage in your learning. Excellent. Someone is saying Oli, Oli, Oli. I haven't heard that in such a long time. Let's have a look. I like your haircut. Yes, those of you that have noticed, I now have a haircut and I also shaved my beard off. I was worried that someone was going to say that they didn't recognize me. No, that is still me. Excellent. Yes, if you want to ask questions, Pele, feel free to ask questions during it and I'll try and reply to you. I'll try and have a look at both of my screens so I can see who's asking what. Thank you. I've had a few people tell me I'm looking fresh today. Love the compliments. In case you are not entirely sure what's going on at the moment then, so essentially these little web classes that I'm putting together, they are mostly there as a bit of a recap. So I'm kind of hoping that this isn't the first time you've come across this, just because I think you might find it quite tricky, but if it is, and hopefully this will be a good founding bit of knowledge for you. So I'm going to go over the key points, I'll go over some exam technique, and you can ask me some questions. Okay, for the next few weeks, as it says at the bottom of this slide as well, this is basically going to be free. So for a couple more weeks, you're going to be able to get these web classes for free. That won't be the case forever, but these ones will be on YouTube for you to come back to if you want to have a look. If you are interested in carrying on with this, SnapRevise 2.0 is going out and it's going, well, lots of people are buying it and it'll be going out on the 16th of October, just so you know. Okay, I'll talk more about this at the end. As with my other webinars that I've done, we're willing to give away one account for SnapRevise 2.0. All you have to do is basically follow us on Instagram and basically tag us, and then we'll choose one random person and you can get your free account. Equally, we've got a coupon code for you guys at the end of this session that you can use to get about 40% off the price. Does this cover globular and fibrous? For the person who's just asked that, yes, it does cover that. Excellent. I'm glad you guys are pleased with what we're doing. Right, so I like to get started then by seeing what you guys already know. Okay, so the first thing I want you to do is have a look at these questions. Start somewhere down here and then work your way up. So these are covering some GCSE points, and then some, well, kind of, and then it's getting more A-level-y as we sort of go up. That is definitely a word, A-level-y. Okay, so start at the bottom, work your way up. Feel free to put your messages on here and I'll start telling you if you are right or wrong. Okay, so the first question is, which molecule is required for a hydrolysis reaction? Lovely, you guys all seem to know that the molecule required is water indeed. Okay, so hydrolysis, hopefully this isn't new. You know that if you want to be breaking something or hydrolyzing it, you need water. Let's look at the next one. So what is the name for the molecules which make up proteins? I'm seeing lots of you saying amino acids, lovely. Someone said VASA earlier, I love that too. So yeah, the the name for the molecules that we need to make proteins are our amino acids. Okay, more about them in a second. What are the names of the enzymes involved in breakdown of proteins? That's one which is also something you should know from GCSE. So Jay Summers, you are saying pepsin, Vanessa protease, Sadie protease, Maha, I don't know if that's how you say your name properly, protease, lovely. Yeah, so the kind of standard we have with enzymes is they always end in ASE, right? If it breaks down a protein, they tend to be called proteases. If it breaks down lipids, lipases, carbohydrates. Okay, so this is a protease. Lovely. Where can you find protein in the body? Who knows this? Dipeptidases too, Lucy, you're spot on. Who knows where I can find protein? In DNA. Good shout. Aisha is kind of like that. In terms of finding it in DNA, your DNA is a bit of this weird molecule where it's very, very long, and it needs to basically wrap around these little proteiny things called histones. So that's why we get this weird sort of coiled structure. So saying that there's protein found in DNA, that is technically wrong, but in the structure that forms DNA, which is called chromatin, you will find some protein. Lovely. Someone's saying ribosomes. You don't really get any proteins in ribosomes. Bone, you get some protein, yes. So you get keratin in bone, you get collagen more in bone. Muscles, that's what I'm looking for. So muscles are the places where you find your protein. Enzymes as well, that was the two that I was sort of looking for. So for those of you who've had your dinner tonight, if you eat meat, then you have probably eaten a muscle of some organism so that you can get protein, because we're not very good at making proteins. Lovely. Hormones, you do get some hormones, true. What roles do proteins play in the body then? So I've sort of said one already. What are some of the roles that proteins play? Proteins are used for building and repairing, as well as to make enzymes and other body chemicals. Yeah, lovely. I like that. Contraction of proteins, lovely. Catabolic, anabolic reactions. So that's talking about breaking things and making things. Don't know if proteins are so much used there. Insulation, not so sure. Hormones, yes. Growth and repair, yes. Energy, possibly. Building muscle, catalase, lovely. So yeah, proteins play. I'm surprised actually that some of you haven't said something which I was actually expecting, but feel free to carry on messaging and I'll see if any of you say the thing I was looking for. But proteins definitely play a role in enzymes. Yes, Freya, I think it's Freya. You've just said it if that is you. So enzymes are the things I was thinking of. Muscles, so contraction, I'll give you that. Someone just said antibodies too. So, or antigens, antibodies. They're all really good examples of where we fundamentally need protein. Okay, so the immune system is very much involved. Mitosis, I can't really think of many proteins being involved in mitosis. It's more collagen, it's more spindle fibers, and you get some contraction, kinesin and dyneins, so maybe not so much. Saying that, Maha, however you say your name, you've just said that proteins are the only ones that contain nitrogen. That's not true because DNA has some nitrogen in it too. Anyway, any other cool little facts that you guys know about proteins? So channel proteins, lovely. I like that you've said that, Charlie. That's really good. Food, yes. Digestion, don't know if that's a fact. Protein is a polymer of amino acids. Perfect. That is really, really good. Used for muscle repair, lovely. So you guys probably know that when you go to the gym, your muscles they're pretty achy at the end. You can literally tear your muscles open and then they get repaired. They tend to get repaired a bit bigger, which is how your muscles grow. Contains hydroxylic and alcohol groups. Okay. They're not the names I would give them, Ahmad, but yes. Nice, you guys know that there are more than one sort of structure. Bold of you to assume I go to the gym. I have a picture of a yellow spot, so it's very hard for me to say very much. Thank you, Hamza. That's very kind of you. Right, anyway. So objectives for today. We are going to describe the structure of amino acids. So we'll start nice and straightforward. Okay, we're then going to look at the formation of polypeptides by condensation. I'm going to make sure that we look at some hydrolysis too. With all of my sort of webinars, I'm going to make sure that you guys can draw this. So I think it's really important that you can draw the condensation of polypeptides. I think if you don't have that, I think you'll find this quite tricky to progress with. Yes, I'm enjoying these comments. These are good so far. Finally, explain how proteins can form secondary and tertiary structures with the example of a fibrous and a globular protein. I'm also going to look at a quaternary structure as well, 2 quaternary structures, just so we're all aware of that. So things I won't cover. There's one exam board that mentions insulin. I'm therefore not going to talk about insulin. I think there's one which mentions keratin, so I'm not going to talk about that either. I don't know why it says insulin twice there, but yes, doubly much. I'm not really going to talk about insulin. So if that is something that you are struggling with in terms of understanding, then I'm going to point you in the direction of our videos. If you look at the one about protein, I'm pretty sure there's something in there about insulin. Okay. Equally, feel free to look at our sort of summary, I don't know, revision page, poster things. Have a look at them. There's some stuff on insulin in there. Jesse, which exam board am I doing? Good question. Here are the spec points for all of the main exam boards that are done in the UK. Okay. So this is the stuff which is mentioned in AQA. So I'm going to cover 95% of this. I'm also not going to cover the Biuret test, this little guy down here. I'm going to do a session on that later. Okay, this is for you guys to look at. So AQA, OCR. I think OCR is the one which has insulin in. Can't remember. Possibly not. edXL as well. So I'm going to do all of them, Jesse. So all of the ones I'm going to cover. Which exam board? Good question. You're going to have a look through here and find it yourself. Because if I'm totally honest, I can't remember. And I don't want to spend my time just going through this. Okay. edXL for biology B is really not specific. Guys, have a look through it. I'm sure you can pause this and find it, but you'll find the one. If your teacher hasn't covered it, then assume that you probably don't need to know it unless your teacher's useless. But having been a teacher, most teachers aren't useless. Most of them are pretty hardworking. Anyway, part one then, guys, is we are going to start looking at amino acids. Okay, so in terms of what amino acids are, we've got these sort of 3 different groups. So you need to know that there is an amino group, and I know for one exam board, it's also called an amine group. So be aware of that. Look up what your exam board calls it, but either an amino group or an amine group. This is this guy over here. Okay, so your amine group or your amino group contains nitrogen, and it contains 2 times hydrogen. Okay, so that's over on my left-hand side. That's nice and straightforward. I've got a hydrogen at the top. Again, straightforward. It's just a nice molecule of hydrogen. And then I've got a carboxyl group. And someone referred to this as something different earlier, and I can't remember who it was, but someone referred to this as something else. I've never seen this called anything but a carboxyl group, okay, or a carboxylic or something, okay. But my carboxyl group contains a double bond to an O, and it contains a single bond to an OH. Okay, so that's my 2 main groups. And then there is this weird R group as well. Does anyone know what an R group is? Has anyone ever heard of this? Let me know. Yes, so someone's just said the right answer. I want to see if a few more people can get there. Alkyl, I haven't heard of that before. Residual group, variable group, rest of the stuff in it. Sharada, nice. And what else are we saying? That's weird. Someone's got the same name as me, which is bizarre. I've just seen Oli V say variable. That's quite concerning, but okay. I love to think there is another person who has my name and spells it the same, but I very much doubt it. It's different, lots more chains, variable, 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 R group, different things. No, different amino acids, side chain. Right, guys, let's clear this up then before anyone else gets confused. So this R group down here, it is just a variable group. Okay, so it's just a variable group, the R group. So I'm going to write that next to it. Okay, essentially, what it means is this variable group can be numerous different things, and that's what makes proteins so diverse. Okay, so the most basic one it can be just a hydrogen, right? I honestly can't remember the name of this. When I was at uni, we had to learn all the different structures of all the 20 amino acids off by heart, and it was annoying. But it could be just as simple as a hydrogen molecule. It could be really complicated and have a benzene ring in it. They can get pretty complicated, okay? But this R group is just a variable region, okay? So I think that this is probably quite important because this is what gives the properties and functions of the amino acid, okay? So it controls the properties of the amino acid. Okay, so let's have a look. Do we need to know any variable variable groups, cricket ray. No, you don't. You just need to know that it exists, right? I could see them trying to trick you by giving you a really ridiculously long R group, which looks really strange, which maybe even looks like a lipid or a carbohydrate. But if it's got this amine group, the amino group, and the carboxyl group, that means it's an amino acid. Glycine, is it glycine, Shannon? Thank you if that is the case. I think glycine sounds right. Glycerin, definitely not. Cool. Right, so let's go a little bit more complicated then. So far, so easy, I'd say. You do need to know this structure as well. I've seen this as an exam question where it's just asked to draw the subunit of a protein or draw an amino acid, and you'll basically get marks if you get both of these sides labeled properly and the R group. Okay, you'll lose a mark if you either mess up one of these or if you mess up one of these. Okay, so be careful with that. In terms of just getting this right, does the R group place matter? This R group could be up here where the hydrogen is, and hydrogen could be swapped. It doesn't make a difference whether that was. Do you need to know cysteine and glycine for the exam? No. Yeah, R and H can be flipped. So polypeptides then are synthesized. So getting back to our sort of vocab, which is really important in biology, we've got this term poly, which means many, and peptide, which means protein. Okay, so polypeptides is basically made up of lots of different amino acids. Okay, in terms of getting the right word, they are synthesized, which means created, in condensation reactions. So condensation gives off water, and I'll draw this out for you in a second just so you can see it. And obviously that involves amino acids. Okay, so if we were writing this as a sort of equation, we would have like 2 times amino acid makes this thing over here. So if you've got 2 of them, does anyone know what it's called? So what's it called if you have 2 peptides? Who can tell me? No? Excellent. Thank you, Abby. So this thing over here is called a dipeptide, right? So you could have some form of word equation where you have, I don't know, 2 amino acids, or amino acid plus amino acid, or I could give you 2 different ones like valine and isoleucine added together, goes to the dipeptide plus H2O. Okay, every time that you make a dipeptide or a polypeptide, whenever you do some form of condensation, it will release some water. Okay, so this one over here, this will release some water too. Okay, as soon as it's got more than 2 amino acids in it, both of these 2 guys, they're called polypeptides. Okay, I don't know if I'm going to be able to fit that in. Maybe I will. Okay, hopefully that's fairly straightforward. I think that's pretty much the same as lipids, where you basically have, what, does anyone remember the names of, yeah, lovely, that person's right, who's just said that. Sorry, that doesn't make any sense. FitCam, again, FitCam, I'm glad you're back. Does anyone remember the name for the 2 subunits that make up lipids, just out of interest? Anyone remember this? Triglyceride is when you've got it. What are the subunits that make it up? Glycerol and gum, not phospholipids. So glycerol and what make up a lipid? Fatty acids, lovely. So if this was proteins, if this was lipids, then this guy over here, that could be a fatty acid. This guy could be glycerol. If you had 3 of these fatty acids, then you'd be making your lipid. Okay, it's always the same. Looking at hydrolysis as well, and I'm going to start making this a bit more complicated because hopefully this is getting really straightforward and boring for you guys because you've done it so much. Hydrolysis is literally the opposite. Okay, hydrolysis, again, looking at our words, so we're getting used to them. Hydro being the word for water, lysis being the word basically mean break down. If you're ever looking for a better word than break down, and you've already used hydrolysis, you could always use something like digest. That tends to be fairly accepted. But hydrolysis meaning breaking down something using water, okay? So to do these reactions, where these sort of little arrows are, I would say you need a little water molecule, so plus H2O. So if I've started off with this polypeptide made of 1, 2, 3, 4 amino acids, water is needed somewhere over here. So you'd end up with something like polypeptides. Sorry for my handwriting, all of those who are people who enjoy nice handwriting. Polypeptide plus water equals, so shown by the arrow, produces amino acids, right? I'm going to write AAs just to save some time. AAs should have an apostrophe, Probably not, should it? Don't know why I did that. Apologies for that apostrophe. So polypeptide plus water makes some form of amino acids. Okay. In terms of what can do this reaction, this is commonly done by these proteases. Okay. So proteases. Ooh, God, this is getting awful, right? You guys are in for a treat. I'm gonna get my little eraser out if I can find it. There you go. You go away. I can do that so much nicer. I can only apologize for the shabby effort that I put into writing so far. So if we have a protease, oh, look how nice and legible that is. If I have a protease, that can be used to help speed up this reaction. Okay, it still requires water, but it will speed it up if I've got a protease. Okay, apologies if it's raining where you are. I don't know if I can have that. Let's have a look. Protease breaks down peptide bond. Lovely. Yes, spot on. So it'll break down the bond, which is here. Because it's an enzyme, and I'll get into this in my next session, I think you need to know about active sites and about things fitting together and being complementary. So look, protease can hydrolyze peptide bonds. Don't worry, I've only learned this for the first time. Oh, I'm glad that you guys are doing this for the first time at the moment. I guess that's quite nice. This should be a nice little recap. Okay. I imagine some of your teachers may have stopped about here, but I want to go a little further. Okay. So if you have a printout of this, if you've managed to download this onto a tablet or something, or you've got this in front of you, I think it's really important you can do this. So in terms of a condensation reaction, let's draw our amino acid first. So I've got a nitrogen, carbon, carbon. I've got my amine group here, or my amino group here with my 2 hydrogens. Let's flip it around. So let's put the hydrogen there, and let's put my R group here to show it doesn't matter. Double bond O, single bond OH. Okay, guys, it's so important that you can remember the names, right? So if you are going to get any marks at all in this exam, you really need to remember this is the amine group, this is the carboxyl group. Okay, so in terms of where my condensation reaction happens, it happens between the amine group and the carboxyl group every single time. Okay, so I've got a nitrogen here, hydrogen, hydrogen, carbon. Let's flip it around again just to be quirky. R, C, double bond O, OH. Okay, lots of you I'm sure know this already, but basically to make water, I'm going to take this OH. I need 1 H from here. Right, I'll take that off, and then essentially what you can do is, if I get my little eraser out, I can basically get rid of this. So hopefully you're seeing this live. Hopefully this isn't going to look confusing, but I can literally remove my water. So if I get rid of my water over here, right, essentially what I'm going to do is you then form a bond between this carbon and this nitrogen. Okay, and this will make some water. Okay, so in terms of this bond, which I haven't really mentioned very much, this bond is called a peptide bond. Okay, so it's called a peptide bond. Okay, so that's the bond here, and it's got the structure of C, double bond O, N, H. Okay, it doesn't matter what way around you have your double bond O and your hydrogen molecule up there, but that is what you're looking for if you're ever looking for a hydrogen bond. Okay, let's have a look at some comments. Let's just see that this is all making sense. Why is the R group on top here? Katie Rose, I've just flipped them just to show that you can. It doesn't really make a difference. How long's the webinar? I'm going to try and make this about 45 minutes from here, or maybe an hour. Tomorrow I have to draw a dipeptide, amino acid, alpha glucose, maltose, triglyceride for a test. Shuffler. This is going to be so easy. I'm envious of you. Let's look. What's on a hydrogen bond? I'll talk about that later. Yeah, same. Lovely, lovely, lovely. Cool. Right. In terms of a hydrolysis reaction, then, again, just to get the practice in of drawing these so many times, right? I'm going to have my nitrogen, carbon, carbon, double bond O. Oh, Christ, what am I doing? Not an OH there, because I've just got rid of that, haven't I? H, H, A. Oh, whoops. Bear with me. There you go. Hydrogen there. Let's get rid of that little thing down there. So we're going to get rid of this guy. We're going to join it up to my nitrogen molecule, carbon, carbon, H, R, double bond O, OH. Right, so in terms of my hydrolysis reaction then, you guys need to know that basically water is going to come in here. So water is going to come into this bond here, And essentially what's going to happen is my carbon on the left-hand side needs to gain an OH, and my nitrogen just needs to gain an H. I've also apparently missed out a H up here too. There you go. So essentially what I'm going to do then in my hydrolysis reaction is I'm going to get rid of this peptide bond. That's now gone. Nice and straightforward. And I'm going to add my, where's it gone? Going to add my OH there, and I'm going to add my H there. Okay, so a hydrolysis reaction is where you basically use water, you break water apart, and you basically separate that bond. Okay, nice. I think you guys are understanding this so far. So let's have a check, friends. How confident are we feeling with this? If you are feeling like this is straightforward, hit me with a 1 or a very happy looking emoji. I feel like I'm in a circle or something. If you're feeling all right, send me a sort of straight face or something like a number 2. If you're really confused, send me a 3 and an unhappy, surprised looking face, and I'll try and explain some of this stuff a little bit more. Okay, tell me how you're feeling. Ira, 4 is not an option. Okay, I'm not allowing that. Okay, mostly 1s. Some people are feeling a little bit confused. Okay, cool. Right, those of you that are a bit confused, I'm going to put an exam question up for everyone else, and I'll start talking over it and talk through it a bit more slowly. Okay, Cricket Ray 0. I'm confused because it's my first time learning it. Okay, fine. That's okay. If it's your first time learning it, I sort of understand it. Guys, here is an exam question then. Okay, here's one that I've found. I don't know what spec this was from, I honestly can't remember, but have a look at it, okay? So it says that biological molecules are held together by a variety of bonds. Okay, if you were in my earlier sessions, you'll know that that means glycosidic bond, which has a structure where you have, can anyone tell me what the glycosidic bond was? I've said it a couple of times and I think a few of you enjoyed what I said. Anyone remember? No? No? Guys, I'm surprised at this. Okay, I think you're probably answering the question. Fine, I'll come back to glycosidic. We looked at ester and we looked at peptide. We'll come and have a look at these again in a second. So let's go through these questions and then, yes, Fatima, COC bond is what I was going for here. Thank you. I feel like I have to say thank you to you a lot. So that's the COC one. Ester bond is very similar, but it's got a double bond to an O. So it's double bond to an O. And then a peptide is the one we've just been looking at, where you have your C, O, N, H, double bond O. Okay, that is right. I hope that's right. Otherwise, I'm very much messed up what I just said I was going to do. Yeah, I think so. See, no, yeah, I did mess it up. God damn. Right. See, that's what happens when you don't think, apparently. Sorry, my bad, before any of you get there. So let's look at these questions then. So we've been told that one of the atoms that make up amino acids has been replaced by the letter X. Okay, state the chemical symbol of the atom represented by the letter X in figure 1.1. So let's have a look. Who got this first one? I'm just going to scroll up and see, because I think lots of you were quite quick here. Lovely, yeah, you guys did all say this. So the chemical symbol of the atom represented by letter X. So be careful here. If they're asking for a chemical symbol, they could be quite harsher and not give it if you said nitrogen, but it should be N. Okay, name the polymer formed from a chain of amino acids. Okay, so the polymer formed, I can't remember if you guys said it. There you go, Tommy Shelby. Tommy Shelby, love it. Lovely, lovely, lovely. You guys are right to say polypeptide. I've just noticed if anyone ever watches this video and they're not bothering to look at the comments, this is going to look like the ramblings of an absolute lunatic, I think, because I'm just saying random names of people and reading out comments, and it must look really strange. I've never actually considered that. Good to know. Probably good to actually think about. Anyway, question 3. Name the bond that is formed when 2 amino acids are joined together. Describe the formation of this bond. Right, this one, I'm going to give you 2 marks worth for it. Okay, so I want to see if we can absolutely get everything right here. So name the bond that is formed, nice and straightforward. So we're going to say that it is a peptide bond. Okay, so we're going to say it's a peptide bond. Nice and straightforward. This second bit, the latter part of this question, is actually a little harder. Describe the formation of this bond. How could we talk about how this bond is formed? What kind of words could we use? Any ideas? Not just peptide. I'll have a drink whilst you guys are contemplating. Lucy, good idea in saying condensation reaction between two amino acids. Yes, I would get the word condensation in there. Condensation reaction. I want a little bit more than that. Where water is removed, yes, I would also, if you say condensation reaction, I guess it's implied water's removed, but let's say it anyway. Let's assume our examiners are really stupid. So water is formed. I want you to tell me where this is happening though. Lovely, Decker is saying it. Decker has said exactly what I want you to say. So it's a condensate, sorry, the name of the bond is a peptide bond formed when there is a condensation reaction forming water, and let's say that this is between the amine group or amino group, whatever you want to call it, the amine group and carboxyl group of two amino acids. Let's write AAs. Okay, don't be lazy like I am in your exam. Never call them AAs, call them amino acids, but just to save some time. That's the kind of thing that they would be looking for. Okay, I don't think they'd want you to say much more than that. They wouldn't want you to say that the carboxyl group loses VOH and the amine group loses just an H. I don't think they'd need that, but they definitely would like you to say this word here, the amine group and the carboxyl group. Thinking back to a student that I've had fairly recently, in terms of where he used to get, I basically do really well and he ended up getting his A star, is he was really, really good at remembering to say things like that last point, right? It's a really easy way to gain so many marks where just a little bit more knowledge can just push you into 2 marks as opposed to 1. Okay, so it's where you have the carboxyl group and the amine group. Just be really careful for that. Cool. Could you say covalently? Because we're breaking the bond, I guess you could say you're breaking a covalent bond. I probably wouldn't. For question I would protein be accepted? I would say probably not. They might allow it, but I would always go polypeptide. I would never risk saying protein because they can find that a bit fussy. How are we looking? Lovely. I think we're all right. I think we're okay with this. So right, let's move on to the next bit then. I think, I don't know why there's the same page twice, so sorry about that. I think that bit that we've just done, I think that is probably the hardest part of this. I think understanding how to draw those diagrams, being able to circle where water is and showing the condensation and hydrolysis reaction, I think that's harder than this stuff, which is just memory recall, if I'm totally honest. Okay, this is a little bit rote learning kind of thing, but it's just one of those things. Okay, so in terms of our proteins, in terms of our polypeptides, we sort of look at them in like 4 separate stages, or 3 separate stages for some of them, 4 separate stages for others. Okay, so we tend to say that proteins have firstly this sort of primary structure. Okay, so a primary structure is like a really, really basic structure. And if you've been doing cells, which I assume quite a lot of you have, when I used to teach us, used to do cells with one teacher and like the biomolecules with another. This is the structure, this primary structure is the structure that your ribosome is making. So those of you that have been doing your cellular stuff and you've been looking at the different organelles, this is the structure that we are forming. Okay, just this one amino acid after another amino acid after another amino acid. Okay, so in terms of what a primary structure is, and I've seen this question a lot, the primary structure is defined as the sequence of amino acids in the protein chain. That is pretty much it. Okay, I'm going to give you a little bit more information just so we can understand it a bit better, but the primary structure is just the sequence of amino acids, one after the other after the other. Okay, for a bit more information, I think it's useful that you guys would know that there are 20 different types of amino acids. Can anyone tell me what's going to be different in each of these amino acids? Can anyone remember? Quick recall. Damn 20, I'll sort of 20, 38. There might be some synthetic ones which we don't really think about. Lovely, it's the R group. Cool, well done guys. That was really quick actually. How many of you are watching them? I feel like there's got to be quite a lot of you to be that quick. I'm impressed. 91 of you, nice. 20 different amino acids, right? And it's the R group which is going to differ for each of these. Okay. Basically, their R groups have very little impact at this stage right now. So in the primary structure, the R group isn't so important, okay? I can't imagine you'd ever get asked a question about this because right now it's not that important. But it is important that we can just define what we mean by a primary structure. So kind of picture this as just the thing that your ribosome has just spat out. So a little bit of mRNA has gone to join it, and then we've spat out all of these amino acids, which have been peptide bonded together. So here are my little peptide bonds, right? And it's just a nice straightforward sequence. Okay, hopefully is there an Oli V version 2? Ugh, guys, lame. You can do so much better than that. Yeah, so that is my primary structure, okay? In terms of my secondary structure, it looks a bit more complicated already. Polypeptide instead of protein in a definition. I would tend to start using polypeptide, yes. What would exam questions be like for different structures? The exam question for this, guys, it will literally be just define this. So define what is meant by a primary structure. That is all I've ever seen on it. So it's a sequence of amino acids in a protein chain. Or go on then, those of you who said it, in a polypeptide chain. In terms of the secondary structure then, we get these two sort of shapes that we start to form. Okay, so the secondary structure involves the formation of beta pleated sheets. And I've seen that AQA have definitely dropped that you need to know these two names off by heart, but I think that's crazy because they're so important. It's the formation of beta pleated sheets, right? Think of like a pleated skirt or something, that sort of a shape it's giving you, and things called alpha helices, right? The reason we get these is due to hydrogen bonds. Okay, so this is due to H+ bonds. I'll talk more about them in a sec. Okay, so the secondary structure involves the formation of hydrogen bonds. Okay, essentially what's going on is I've got a weird little interaction between the hydrogen group, which as you can see, I don't know why I've scribbled it in so much. Yeah, I've scribbled it in so much you can't really see it anymore, which is on the amine group. And that's going to have some form of reaction with the oxygen, which is on the carboxyl group. Okay, so I get these weird little interactions forming these things called hydrogen bonds. Okay, basically what happens is, as opposed to being just a nice 2D sort of flat structure like my arm is currently vaguely 2D, of just amino acid. Essentially what's going on is because of these hydrogen bonds, it will start to curl up a bit. Right, so it starts to curl up either into these alpha helices, and these guys are really important in terms of channel proteins in your membranes. And your alpha helices, which you just get in basically every enzyme at some point or another. Okay, so yeah, you get these kind of coils. Very helpful writing three letter words. Me? No, possibly not. Cool, I'll just ignore that comment. Why are they forming? They will just naturally form due to hydrogen bonds. I'll tell you more about hydrogen bonds in a second. I won't give you much, but a bit. So the secondary structure due to hydrogen bonds, that's what H+ is. I'd always write hydrogen as well. Try not to be too lazy when you write stuff down in biology. And that forms beta pleated sheets and alpha helices. I get a tertiary structure too. So this is where it all goes a little bit wild. So my nice, and I say wild, I mean wild for biology, and that's probably not that wild. My tertiary structure is the overall 3D structure of a protein, or just to make everyone happy, let's say polypeptide. I think I prefer polypeptide. Okay, so the tertiary structure is the overall 3D structure. This is almost like the final structure, unless a protein is made from more than one polypeptide, and I'll talk about that in a second. Okay, so essentially what you have here is you have lots of these alpha helices. So I'll have a lot of alpha helices. And the reason it's helices is because it's helix, and that's the plural for helix, in case you didn't know. Same with matrices. There'll be lots of beta pleated sheets forming, right? And you need to know that that's due to these hydrogen bonds. But then we get these other bonds happening too. Does anyone know what the other bonds are? Does anyone know this? Who can tell me? Trisha, I'll get back to your question in a second. Aisha saying that year 12 stuff's really easy compared to year 13 stuff. You'll feel that way when you go to university if you do anything, but everything you did at A-level was really easy. Lovely, guys. Smashing it. So bonding, we get these things called disulfide bridges, right? I would try and get in the habit of spelling sulfide with an F as opposed to a PH, because that's what everyone does. It's only the English you spell it with a PH. So disulfide bridges, okay, they form between two amino acids called cysteine, which have sulfur in them, and they form a weird covalent bond. So disulfide bridges, someone said ionic bonds. So you guys, I'm assuming, all did some form of chemistry when you did your GCSEs. Ionic bonds just being a sharing of electrons. Let's see, van der Waals forces, kind of, not so much really. There's hydrophobic dash hydrophilic interactions. Okay, that is a confusing one. Essentially, what it means is all of the parts that are hydrophobic, which hate water, they all kind of bunch up on the inside. All the bits which are hydrophilic, they sort of go to the outside. Someone did ask a good question earlier, and I've forgotten what your name was. They basically said, how do you know if it forms an alpha helix? Sorry, an alpha helix or a beta-beta sheet. It just depends on where the hydrogen bonds are forming, basically, which will somewhat depend on the shape of the amino acids. Will this be the R group? Yes, perfect. So all of this is determined by the R group. So all of these things here, let's put this in like a big old bubble to make it stand out. There are some nice little lines coming off of it to show just how important this is. All of these things that occur, these are all due to the R group. So the overall 3D structure of my protein fundamentally is formed due to this variable group in my protein, in my amino acid, right? All of that comes down to this R group. Okay, disulfide bridges, I'll get to that in a second. Covalent bonds too, yes, sorry, my bad. Covalent bonds, ooh, getting too close to the bottom. Covalent bonds will also form here as well. Well done, whoever said that. Yes, so Oli Vaughan is correct. God, that feels weird to say. Polar and non-polar. Polar basically means hydrophilic. Non-polar means hydrophobic. Yeah, so Abby, you're right. It is the sequence of amino acids, but it's the R groups on those amino acids that causes this to actually happen. I'm not praising myself. There are 2 people called Oli Vaughan, or 3 of us now, and I'm not praising myself, Hamza, so shush. But that is the tertiary structure. Okay, so it's really, really important that we're sort of linking all these ideas together. So I've got a primary structure consisting of just my sequence of amino acids. And this is often an exam question too, to ask about how the structure of proteins is sort of broken down. You'd say there's a primary structure, which is the sequence of amino acids, a secondary structure due to hydrogen bonds forming these alpha helices and the beta-beta sheets. And then finally, my tertiary structure, which is the overall 3D structure, which is formed due to the variable groups found on my amino acids. Okay, that forms disulfide bridges, ionic bonds, hydrophobic / hydrophilic interactions, and covalent bonds. Okay, so confusing. Right, I'm just going to ignore this from now on. Otherwise, I think I'll spend my whole time just sort of going through this weird meta argument in my head, which no one wants that. Sorry, final thing then is we can also get this thing called a quaternary structure. Okay, this is just one of those other words, you know, after primary, secondary. If you go to university, that's considered tertiary education, as is college sometimes. Quaternary is just the next one up, right? This describes how multiple polypeptide chains are arranged to give a 3D structure. So the key example here being something like hemoglobin. Hemoglobin isn't made of just one polypeptide. It's actually made of 4 chains, right? So the quaternary structure is where you have more than one polypeptide chain, and you also have these things called prosthetic groups. Okay, I've seen this written in one exam board as a non-protein group. Does anyone know what I'm talking about when I say a non-protein group? Does anyone know the example that I'm talking about here? Can anyone tell me? Someone said prosthetic groups. Love that. No, Oli Vaughan doesn't know. Yeah, I do. No. A group with no proteins. Yeah, so Trisha, you've smashed it. So the group that I'm talking about, the main prosthetic group you're ever going to come across in the next couple of years is called heme, right? Which is basically an iron group. So the thing that is what hemoglobin is named after is this weird little molecule, which is, I can't remember if it's iron 2+ or iron 3+, but it's one of them. But basically you find it embedded in this structure, like here and here. This one isn't, this isn't a hemoglobin, but let's just pretend that it is, right? You get these weird non-protein prosthetic groups, and that is my quaternary structure. Okay, You're also going to find a whole lot of ionic bonds in here as well, which holds it together. So there'll be some ionic bonds holding your quaternary structure, and you'll also get some disulfide bridges as well, holding all of this together. Okay, so I think I possibly have an exam question on this. I do in one sec. So let's see, what are you saying? So iron and hemoglobin, nice. Glycoproteins contain a carbohydrate part. There you go. True, but they're not strictly completely proteins. What about histones? Are they a prosthetic group? No, they actually are made of protein. Fe2 plus, Giso, I'll take what you say because I honestly can't remember. Collagen is quaternary structure, but it doesn't have a prosthetic group. No, good point. So a quaternary structure, it doesn't have to have a prosthetic group. It's just some of them do. I think by definition, they have to be multiple polypeptides though. Okay, so the quaternary structure has to have more than one polypeptide. They may or may not have a prosthetic group. Aisha, I will go over 2, 3, and 4 again. So, gone for Aisha's sake, right? The primary structure then is just the sequence of amino acids, straightforward. The secondary structure, basically, because of hydrogen bonds, we get these weird shapes forming. So as opposed to being nice and flat, it forms this sort of crinkled old mess of stuff with some spirals in it, which are what alpha helices are, and beta pleated sheets, where you basically get the hydrogen, your amino acid chain sort of doing that. So that's due to hydrogen bonds. Tertiary structure, you have a whole load of other bonds that curl it even tighter. So it starts off nice and straight like my pen. It gets slightly crumpled and crinkled because of hydrogen bonds. It gets even more crinkled by all these other bonds. And that's what gives you this protein, which has this such specific shape. So those of you that are familiar with enzymes and you're familiar with the active site, you probably are aware of how specific it has to be, right? You have this lock and key mechanism where you have a perfect lock and you have a perfect key. And if they come together, very like, I don't know, Kung Fu movie there. If they come perfectly together, then it can be, you can have this reaction happen where you basically will hydrolyze whatever my substrate is, right? If you don't have this perfect folding of amino acids, like due to these ionic bonds, these hydrophobic, hydrophilic interactions, then your enzyme just won't work. Okay. Induced fit. Yeah, Giso, you're right that there is an induced fit theory too. I'll get to that another time. Apparently lock and key is a lie. Yeah, that is a lie. I was trying to make it a little bit easier. Induced fit basically is where something will change shape. So it doesn't fit perfectly and then it changes shape. I do know that. Right, so just so we're all aware, and I like to tell my students this just to make life a little easier. You don't really need to know too much about this. This is more chemistry. But just so we all know what a hydrogen bond is and why it happens, essentially, if you're not aware, hydrogen molecules are very slightly positive, right? And we give them this weird little symbol, which is a little D sort of thing with a little curly tail. And oxygen is just about negative. And this is because electrons like to sit a little bit closer to oxygen. In this bond, in this covalent bond, you guys will be aware that in covalent bonds, hopefully you have these shared electrons where they sort of share electrons together. Because these electrons sit a little closer to oxygen, I end up with this very slightly negative oxygen and a very slightly positive hydrogen. And that's what forms this hydrogen bond to form. Okay, so a hydrogen bonds form because you've got a very slightly negative oxygen and a very slightly positive hydrogen. Okay, just so you're aware as well, you can also form them between nitrogen. So it can be between hydrogen and nitrogen as well, as opposed to hydrogen and oxygen, just so we're okay. Yeah, you're right that that is a delta symbol. Yes, this is based on electronegativity. Love that some of you are doing chemistry. Okay, this is why certain things are polar as well, which someone said. In terms of ionic bonds, I'm not going to spend too much time here. Basically, you guys hopefully have some recollection from chemistry that some things, if they don't have a slight negative or a slight positive, they are just naturally positive, and other things are just naturally negative. That means that there is this electrostatic attraction. Okay, that's one of the key forces as well that we come across in physics. So whenever you got taught about like all these different forces at GCSE, it's a bit of a lie. There's only really like 3 or 4 forces that we ever talk about, but electrostatic is one of them. Ionic bonding, that's when you have oppositely charged ions, they stick together. Does this have a link to Zwitter ions? I honestly can't remember, Cricket Ray. It's been a while since I've done chemistry at A-level. If we look at disulfide bridges, essentially what these are, or disulfide, yeah, just so you see this as well, these are sometimes called bridges as opposed to bonds. I tend to use them interchangeably. But essentially what's going on in this picture is you have your little sulfur in some R groups. So some of these R groups, if you look back at your structure, some of these R groups contain a sulfur molecule. And sulfur's a bit weird because sulfur can form a covalent bond between another sulfur. I can't remember how many electrons are on the outer shell of sulfur, but it just means they can share. So a disulfide bridge is just this weird bond between two sulfurs. Someone's internet died, and yet they just messaged, which is interesting. People with lots of disulfide bonds have curly hair. I've never heard that B1324, but interesting if that is true. Here's what I said earlier. Look, if you've got a hydrophobic region, they tend to stick in the middle. Hydrophobic, phobia, think of arachnophobia, they hate water. They stick in the middle. All the nice hydrophilic bits, they like to stay around the outside, so they're constantly in water. So that's what a hydrophilic interaction is. You'd never get asked about this, guys, but just so we're all clear on what these things are, I think it's important. So this, yeah, for any of the examples, I don't think you need to know that from what I seem to remember, unless one of them possibly mentions it, but I've covered it anyway. Cysteine is just an amino acid, Khadija. So guys, tell me how you're feeling then. If you're finding this straightforward, hit me up with a nice smiley face. If you are feeling all right, maybe not, send me one of these ones or two. If you're confused, send me this guy. He's going to have a little tear. Look at that. Tell me how you're feeling with this so far. Good for essays. Yeah, this information is good for essays. That is an actually amazing point, Charlie. So Charlie's just said that information I've just told you about will be good for essays. Because it's not specifically on the specification, you will get bonus marks if you can say things like that, just so you know. Lovely. 1.5, I'll take that. Louise Ion or Louise Lon, whatever your name is, that sounds quite cool. Synoptic essay is correct. Louise, hopefully this will make a bit more sense in a second. Okay, so here's another exam question to have a look at then, guys, and I'll talk through this in a second, and then we'll do the final bit looking at collagen and hemoglobin. So describe how, so each amino acid has a different R group, as you guys are aware. So there are 20 of them, as I said. Describe how these R groups can interact to determine the tertiary structure of a protein. Okay, tell me what you would say about this. So here is a nice question where you can say a whole lot of stuff. So what kind of things would we talk about? What would you say? UK drill analyzer. Interesting. It is ion. No, not ion. Lon. Sad. Guys, lovely. So someone has said something already. So if you were to mention in this question hydrogen bonds, you would probably be getting a mark. Okay, so hydrogen bonds. Because we're A-level students, let's make sure we're saying everything in as much detail. Hydrogen bonds between O and H or N and H. Okay, lovely. Lovely. Lucy has just covered everything for us, basically. So describe how these R groups can interact to determine tertiary structure. So we could say that some R groups lead to, let's say, disulfide bonds. If you know it's cysteine, that'd be good. We could say the same thing here. Some R groups lead to hydrophobic / philic interactions. Oh, that's terrible, sorry. Philic interactions, I'm just going to write inter. You guys know what to say. Interactions, we could say about ionic bonds. Have I missed any? Probably. Different amounts of alpha helices and beta pleated sheets. That's due to the hydrogen bonds. So I guess so, but this is more talking about the R groups. So it's more talking about the tertiary structure. To these R groups interact with some tertiary structure. Lovely. I think the only thing you'd want to say towards the end of this little question, covalent bonds. Oh yeah, smash it. Thank you. Covalent bonds too. The only thing you'd want to say is that these R groups, therefore, can cause things to come closer together, causing the structure to curve in more, or it can cause things to sort of repel each other. So in the sense of hydrophobic / philic interactions, that can cause certain things to sort of move away from each other, which is going to make it flatten out again. Okay, so I think you'd want to raise the point at the end of this question that R groups, so variable groups on the amino acid lead to attraction or repulsion. And then you've sort of given your reasons above. Okay, repulsion always seems like a sad word, I think. But yeah, so that's the kind of thing that you want to be talking about. Okay, I think that'd be quite a nice question. I haven't seen that come up for a long time or anything like that, but I think that'd be quite a nice question. Okay, final part then that we're going to look at is going to be collagen and hemoglobin. So in terms of these two guys, I did say that you also, for some boards, need to know a little bit about insulin, but you tend to just need to know these two. And to be totally honest, hemoglobin, this one I wouldn't worry about so much because it comes up in so much detail later on. So collagen is the only one where you need to know a few little facts about it, and then you're pretty much it. Can you bullet point an answer in A level? Yes, so you can bullet point it for most of your questions, and they won't take points off, but not your synoptic essay. Okay. Nice. I've just seen someone else. Abby, that's really good. So just so we know what the function of collagen is then, collagen is this really odd little protein where it's got this weird little coil in sort of making it up, right? It's this weird little coil where it sort of spins around like a helix. I'll tell you more about that in a second. But then you always have three of them, which also helix around each other. So you end up with this weird name, which is a triple helix, which you don't really hear very often. But the function of collagen, and you find it in your connective tissue. So in biology, in mammalian like histology, which is the study of tissues, you have fibrous tissue, which has lots of collagen in it. You have epithelial tissue, and you have muscular tissue, and you have nervous tissue, right? But this is one of the main things that makes up one of your main tissues that you have, right? So collagen is what you have that basically makes this really strong stuff that you find in your bones and your tendons. So you need to know that collagen is there to provide strength. So provide strength to, let's say, bones, tendons, and skin, right? That is why you have collagen. Okay, so it's providing lots of strength. And the way that it does that is due to this structure. Okay, so the way it does that is it's made of 3 polypeptides. Okay, out of interest, if it's got 3 polypeptides, what is the overall 3D structure that we would say for it? So what is the overall 3D structure that it has? Can anyone tell me? Collagen is a type of protein, Cricket Ray. Tripeptide, no, quaternary, Brandon, yeah, exactly what I'm looking for. So the structure is 3 polypeptides. So it has this 3D sort of quaternary structure, which I'm now terrified that I can't spell quaternary. Okay, so it has this quaternary structure. This forms something called a triple helix, not just a regular old double helix, but it forms a triple helix. Love that word. Essentially, the reason you get these 3 stuck together is between each of them, you have lots of hydrogen bonds. So collagen is formed due to hydrogen bonds. And then in a very similar way to cellulose, so it's hydrogen bonded together. In a similar way to cellulose, you end up forming things called fibrils, which are like long chains, and things made of lots of fibrils, which are called microfibrils. Okay, if you Google what a picture of collagen looks like, you'll see where they tend to show you it as like one single strand, which will be just one sort of bit of collagen, and then they show it wrapped around. So you see this triple helix structure, and then I'll show a fibril where you have loads of them stuck together, and then you also show, a microfibril, and then I'll show you this fibril, which is made of absolutely loads of them. Okay, so, oh, and we also get fibers of this too. Okay, so what are we saying? Yes, parallel cross linkages. That is a thing that connects this purple guy here to this green guy here to this green teal guy here, right? So we get these sort of cross bridges that form between them. Okay, so in terms of this protein, this is called a fibrous or fibrous protein. Okay, it's always a good example of a fibrous protein. Another one would be keratin, which is the stuff that you find in your fingernails and stuff. This stuff here as well, this is the most abundant protein in your body. Almost, I think it's like 25 to about 35% of your protein is collagen. So you have more of this than any enzyme, like by far. Okay, so that is everything you really need to know about collagen. So it has this high tensile strength. It's very, very strong because of these 3 alpha helices. It has a large number of hydrogen bonds between the helices, and the collagen molecules are combined together to form fibrils, fibers, microfibrils, those sorts of things. Okay, I think for AQA you don't need to know this in much detail at all, but I'm pretty sure for all of these you don't really need to know this in that much detail, so I wouldn't worry too much, and I'm definitely going to come back to these in the future anyway. Last thing I'm going to talk about then, and then I'm going to start because it's been an hour now and I don't want to take all of your evening up, because I'm sure you have better things to be doing. In terms of hemoglobin, does anyone know what hemoglobin does? Does anyone know what hemoglobin does? It's this thing which is talked about all the time. It looks like these guys have little moustaches. That's nice, isn't it? Does anyone know what hemoglobin does? Carries oxygen. Yes, spot on. So the function of this is it will basically move oxygen from one place to another. You'll find out loads about hemoglobin this year. It's really cool. I like this topic. And you basically have this thing called loading, where it takes oxygen in and then it unloads. Brilliant. And it does that by changing its shape. So it very specifically changes its shape, which allows it to get rid of oxygen or take it in. Right, and that's caused by the conditions that surround it. Okay, the structure, yes, lots of you are saying it. So it's got a quaternary structure. Okay, this quaternary structure is made by 4 times polypeptides. Okay, you have 2 different types. So let's say that's one polypeptide, that's another polypeptide, that's another polypeptide, that's another polypeptide. These 2 are technically the same. So despite that there are 2 of them, but they are the same. So there are 2 different types, but there are 4 of them in total. Okay, each one has a heme group within it as well. Okay, so basically hemoglobin is something that transports oxygen around, as I was saying. It's got this prosthetic group, this heme group, which contains iron, which combines oxygen. And fortunately, it has 4 of these molecules of heme, so it can carry 4 oxygen molecules around at any time. So look, are all globular proteins quaternary? I don't think so. I very much doubt that all of them are. I'm sure there are exceptions. Lots of them are. Are you going to do a live stream on HUT? My plan is to do a live stream on all of A-level biology by the end of this year. So I'm doing year 12 and year 13. So yeah, I'm planning to do all of them, so you know. Enzymes are globular and tertiary. Yes, lovely. Right, guys. I'm not going to do that. I'm going to skip. So because I want to finish this on time, you never answered my question. I did answer your question, Trish. Yeah? I'm pretty sure I did anyway. The alpha helices and the beta-beta sheets were hydrogen bonded, and it's kind of a bit random, and it depends where your amino acids are, essentially. Anyway, no more talking about hemoglobin. I will get to that, or you'll definitely come to it in a lot more detail than what you can do now in 2 minutes. Okay, right, guys, just to have a go then, explain some stuff. In terms of SnapRevise, and hang around if you want this code as well. In terms of SnapRevise, just so you're aware, this is coming on the 16th of October. Okay, so not very long. What's the date today? Should we know that? 30th September. So like 16 days, SnapRevise 2.0 will be up and ready for you guys to have a look at it. Okay, I'm discard this. I'll show you. Look, here it is. This will try and delete itself and hide itself. Hang on. Here you go. So if you go onto our website and go onto the SnapRevise 2.0 logo, here you go. This is what we've got that we're offering you. So we've got loads of topics. You can pick any topic you like in biology. Obviously, you can't do history or something. Chemistry you can do as well. We'll give you a quick little test to see what you know. You can then fill your gaps. So we've got loads of videos. If you have a YouTube open right now, don't you? Obviously, that's done. Go have a look on our website and have a look at all our videos, because there are millions of them. Not millions, I probably shouldn't lie. There are literally hundreds of them though. But yeah, you can watch some videos, and we got some cool smart tech which will give you the right video so you can see what you don't know, be it the bronchioles, the alveoli, or whatever. There'll be some self-marking little quizzes, so you can do loads of quizzes. There are thousands of questions on that. I mentioned this earlier. If you're looking for insulin, you don't know much about it, you can get our nice little revision guide. You can download them, put them around your room, get people to test you on them. It's basically all your revision notes for you. There are some walkthrough videos for exam questions, even more exam questions, which you can define by difficulty. There are some nice points you can get. And then finally, most important bit in my mind is you have people like me who will be doing sessions throughout the year where you can basically ask us questions and I will try to respond to them and tell you basically some good answers. So the plan is, I believe, that you'll have there'll be times of the week where you can ask questions. I'll be online. I'll just reply to you and try and explain it as best I can via typing it. It probably will involve lots of little diagrams too. Okay, that's what SnapRevise is. So that's what's coming in 16 days, just so you know. In terms of what we're offering as well, there are 3 packages. There's the basic one. If you've signed up already, you will already have this basic one. You'll have a discount for the pro one. You'll have a discount for the ultimate one. Key thing is if you want these web classes, that is in our ultimate package. Okay, I can't get enough of saying ultimate package. I find that really funny every time I say it. Okay, there you go. Again, look, guys, have a look. Oh, I keep having to discard my stuff. Let's discard it. As I mentioned, if you go on our YouTube accounts, here you go. Here is the web class you are currently in. Apparently only one person's waiting. Don't believe that. Look, there's one tomorrow about moles. I'm sure lots of you want to know more about moles. You can also see some of our older videos. If I go on this, I'm sure it's going to start playing something, which will probably maybe not. There you go. Here are some of our other videos. So if you missed my earlier sessions on photosynthesis or lipids or whatever else, you can find them. Look how many there are. When I said there are hundreds of them, I really do mean there are hundreds of them. This covers pretty much all of A-level biology. I would literally be using this all the time if I didn't get something. Okay, so have a look through that. And then finally, last little thing then, here is the discount code for those of you that have waited patiently for that and nothing else. There is your discount code. And finally, I can breathe. Hopefully by now, you guys should know the structure of amino acids. So you should know the N, C, C, R group, H, amine group on my left-hand side, and carboxyl group on my right-hand side. You should know the levels of structure in proteins. So primary, which is given this kind of like annotation 1 with that little symbol, which means prime, 2 prime, secondary, tertiary, 3 prime, quaternary, 4 prime, and then hopefully how the structure of hemoglobin and collagen relate to their function. So how hemoglobin has these 4 heme groups, which allows it to move lots of oxygen around, and how collagen has all these hydrogen bonds and this triple helix structure, which makes it really, really strong. Okay, guys, I'm going to give you a few minutes to ask me any questions, which I'll have a go at doing, and then I am going to head off and enjoy my evening as well. So any questions, have a go at asking them now. Let's have a look. Someone's asked something complicated. Describe the difference between the HbA allele and the HbS allele and state how this difference affects the beta globulin polypeptide. And that question is far too complicated, and I think I'd need a lot more information to answer that. Explain the synoptic essay. Yes, my pleasure. Essentially, this is the best way for you guys to make marks as well. In the past, my students have always done best on it. At the end of paper 3 in AQA, you'll get this question, which is, I think it's 25 marks. Yeah, it's definitely 25 marks. And it will give you 2 options, and it will say something like, explain how ions are important in biology, / one of the best questions I've ever seen, how are nitrogen-containing compounds important in biology? You basically have to write at least 4 paragraphs about when that's important in biology. So nitrogen-containing compounds, talk about DNA, RNA, any enzyme, anything like that. And you talk about it, and you get lots of marks. And it's fairly straightforward. Where is the hydrogen peptide bond for a polypeptide? So the peptide bond is, if I draw another one, so if I have my H there, my H there, this is my peptide bond, which will form between my my amine group and my carboxyl group. So you take away 2 H's, H2O, and you form this nice bond here instead. So I'll get rid of that. There you go, that's now gone. That is my peptide bond. In terms of my hydrogen bonding, essentially the way you've got to think about it is if I had another amino acid somewhere down here. So let's just draw the basic outline of it, C, N, C, C, right? Essentially, you could have a hydrogen bond forming, if I've got a hydrogen here, between this O and this hydrogen, right? So this one is slightly negative, delta negative. This one is slightly positive, so we form this interaction. No synoptic essay in OCRA, which I'm glad of. Jisel, you say that, but they're not actually too hard. Can I write more than 200 words? So I'm sending it in 2 parts. Oh, right, sorry, Leonide. I see. Feel free to have a go. I'll stick around to try and have a look at it. Never been more happy to be shushed by the great Oli. Weird. Thank you. I'm glad you like that. Which two substances are formed when two amino acid molecules join together? So two amino acid molecules join together, you're going to form a dipeptide and you're going to form water. How often do you do your live streams on both year 12 and year 13? At the moment, so I think the plan is ultimately for me to be doing one pretty much every day, but I'm doing one on Monday for year 12 and one on Wednesday for year 13. That's what my plan is for the next few weeks. When is hydrolysis and condensation actually occurring? Every metabolic reaction that happens in your body involves hydrolysis and condensation. Every time anything is digested, you have a bit of that going on. So literally all the time. Leonides, let's have a look at your question. So polypeptide synthesis occurs, oh God, you are sending loads of this. Okay, so you got an H, I hope I'm not doing your homework for you, Leonide. You got a describe the difference between an HbA allele and a HbS allele and state how this difference affects the beta globin polypeptide, the hemoglobin molecule. Polypeptide synthesis occurs before a red blood cell is released into circulation. The HBB gene codes for globin, polypeptide of hemoglobin. There are two alleles, HBB, known as HbA and HbS. Describe the difference between HbA allele and HbS allele and say the difference effects. God, if I'm totally honest, Leonide, I don't know. I don't know if I'm going to be able to answer this right now. So polypeptide synthesis occurs where a red blood cell is released in circulation. I guess you want to talk about probably the, I think this is probably easier than you think it is, Leonide. I think this question is probably just asking you that if you've got an allele difference, if you've got a change in your allele, which is basically your gene, then you're going to change your DNA sequence, which means you'll change your mRNA sequence, which means that you'll get different amino acids in the sequence. So by doing that, you're changing the primary structure of that protein. So in terms of the differences, I'd say you'd have a different primary structure because you've got different alleles, which means you'll get different hydrogen bonding. So you have different secondary structure, different alpha and beta helices, and then different tertiary structure, and mention all the other tertiary structure things. I think that's pretty much how you're going to get your marks on that question, if it were me. Anyone else? I guess I'll be seeing you on Wednesday then. Oh, I'm pleased you'll be seeing me on Wednesday, yes. Chemist content be applied to OCR all day. Yeah, guys, all of this content, I'm making these lessons so that they cover all of the boards, right? I don't want any of you to feel like, oh, I'm not covering your stuff or I'm not covering your stuff. I'm trying to make it cover everything when I'm taking out a few little bits that I think aren't too relevant that you can sort of research yourself. Can you please go through hemoglobin exam question at the end? Yes, Fatima, for you, I can. Hang on, let's look at some of these others. How are 4 polypeptide chains and hemoglobin bonded together? Hydrogen bonds, all the other bonds that we said. How can you use a bioreactor test to distinguish a solution of enzyme lactose from a solution of lactose? If it's bioreactor, basically changes colour to purple, so it wouldn't do that in the lactose one. Is this live on the Heart for free? I don't know if it's going to be free by the time I get to the Heart stuff. I guess it's going to come, we'll have a look at that point in the future. Safe man, am I going to watch the Arsenal match? I don't really care about football, if I'm totally honest. I'm more of a rugby man. Right, so yes, Fatima. So state one function of hemoglobin. I think that one's really easy. So that one's just transport oxygen. This can be the last thing, guys, as well, by the way. When do you do... Apparently I said that, I mean, I've read your question because I want to answer you. When do you know when to use mutations in a question? If you've got a different allele, there's been a mutation at some point, Zaki B. So hemoglobin contains a prosthetic group known as heme. Collagen does not contain a prosthetic group. Describe 3 other ways in which the structure of hemoglobin differs from that of collagen. Okay, so we would need to say that hemoglobin is globular versus fibrous, right? You'd need to not be lazy like I'm being. You need to say that this is hemoglobin, which is often given that little symbol. And this is collagen. So that'd be your first mark. You'd want to say that the subunits for the two are different. So for hemoglobin, we have 2 different subunits. So there's an alpha and a beta sort of subunit, right? Whereas collagen is just the same thing repeated over and over again. So that's in hemoglobin. And then finally, you'd want to say that hemoglobin is made of 4 different, or four molecules of protein. Don't know if molecules are the right word I'm going for. Let's say, let's change that. Let's say 4 polypeptides whereas collagen, collagen is made of, and then you could sort of argue either way. You could say it's made of one, because that's the fundamental basic part of collagen, but it comes together to form this triple helix. Collagen is made of one, which formed the triple helix. Okay. Lovely, guys. You know how to answer this question anyway. Who will win Rugby World Cup? Tokyo will win. Tokyo aren't playing in Rugby World Cup. Japan are. I don't know. Probably the All Blacks, I would imagine. Wales played well the other day, which was pretty disappointing. I sort of wanted Australia to win. Anyway, your message just got deleted. Guys, I'm going to head off now. So hopefully this has been a good session. Hopefully you know a little bit more about proteins. Hopefully if you're the person who had an exam on this the other day, you slightly know this a little bit better. My second point here, 2 different subunits in hemoglobin. So hemoglobin has the alpha and the beta, whereas collagen only has one type of thing that makes it. Anyway, guys, I'm going to head off. So I hope you have a really nice rest of your week. If you want to do some year 13 stuff, and if you are year 13, I'll see you there. If you're in year 12, possibly don't, but up to you. Otherwise, I'm going to sign off. So have a really nice week, and I will see you again at some point in the near future. See you later.